Now, for lecture 1.4, we're going to be discussing antibodies. Antibodies are part of our humoral immunity because remember these are what are produced by the plasma cells, the differentiated, clonal expanded B cells. There are 5 types of antibodies, what they call classes or isotypes. IgA, IgE, IgG, IgM, and IgD. Remember I told you that Ig stands for immunoglobulin, immunoglobulin class A, immunoglobulin class G, and so on. Now, all of these have at their basis the same structure. The constant region, the variable region, the light chain, heavy chain, is just how they are arranged. May vary from class to class. Now, whatever class they are, they have the same roles. Protection. Protection via neutralization, opsonization, or activating another serum protein known as complement. By neutralizing, they will bind up the toxin. The different variable regions from different antibodies will bind to the toxin and they will hold it in suspension. For a toxin to work, it has to come into contact with a cell, has to come into a contact with a receptor on a cell. By being bound up, held in suspension here, as it's shown here, well, what if you have the bacteria that's actually in the bloodstream? Antibodies there can bind to it. Doesn't need to be as many because instead of opsonizing, just opsonizing, what they're going to do is they're going to begin a cascade of proteins that will form tubes, holes, punched holes through the surface of the cell of the bacteria. Everything inside the cell, as it shows here, leaks out. Cell dies. The Fc portion of the antibody binds to the receptor. Bacteria gets taken up. All of this that's being spewed out will be recognized as non-self, acting as cytokine, acting to activate other cells, more cytokines, more chemokines, more things will come. Now, when it comes to T cells and B cells, they need each other. Okay, one cannot be activated without the other. They have a yin-yang interaction. T cells, once activated, will influence activating other cells to upregulate at the initial response of an infection, and then their influence will come in later when they tone it down. There are 4 main types of these helper T cells, the ones that are going to activate. These are going to be the helpers. They help other cells become activated. Notice here, TH1, TH2, TH17, TFH cells. The subscript H denotes helper, so it's a T helper cell. Type 1, type 2, 17, and follicular. Why does it jump from 2 to 17? I don't know. It has to go with how the cells were identified and then later genetically tested to show that they were actually different lineages of T cells. And then you have the killers, the ones that hunt down and find infected cells. Infected cells that are expressing a non-U epitope in their MHC, which need to be told to die. Cyto means cell, toxic means killer. So cytotoxic T cells are actually cell killer cells. Because remember, we talked about this. A virus, again, things are not drawn to scale here. A virus, one goes in, gets inside, takes over. The surface of the cell will become altered. These viral proteins and things that are being made that are now on the surface, some of those are going to be digested up and they're going to be expressed on MHC, which is not showing right here. Along comes cytotoxic T cells. the cell killers that have been told there is a virus infection. They will recognize these non-self viral proteins. They will bind, T cell receptor will bind to it. Primary signal, secondary signal comes through. You see here the nucleus has now chopped itself up. That's the first sign of apoptosis, cell suicide. Some of the times it's going to be because there is a bacteria. Here, Mycobacterium, could be Mycobacterium tuberculosis, Mycobacterium avium, Mycobacterium leprae, gets inside the cell and then it hijacks the cell. Remember, things that get phagocytized should be digested with a lysosome. Well, Mycobacterium is one of those types of bacteria, hijacks the signals, and it now gets to reside inside a vacuole. All the cytoplasmic contents will now be pumped into the vacuole for the bacteria to go to utilize metabolically. So it's now protected in here from all the rest of the immune system of this host. Well, this is going to cause the cell some consternation. It's going to make it change metabolically. Some of these mycobacterium are going to die. They're going to be digested away, broken down, and their epitopes, their antigens, are going to be presented on the surface. Cytotoxic T cell or a Th1 cell comes in, not a cytotoxic, a Th1 cell comes in, recognizes that this shouldn't be, and it's going to tell this macrophage to turn on other genes, to turn on other signal pathways that it doesn't normally use. By using these other pathways that it doesn't normally use, it can overcome the mycobacterium, halting of its signals, and digest it away. Why doesn't it just do this from the get-go? Well, because it's overkill. If the macrophage cannot tell the difference between E. coli and Mycobacterium. E. coli, easy to kill. So it's going to have certain pathways that are set up so it can kill E. coli, staph, all these other bacteria relatively easily with minimal waste. Well, this is a very expensive process metabolically, so it needs to wait until it's told to class 1 CD8, class 2 CD4. So what happens in a nutshell? Okay, we'll be going over this in more detail in later chapters, but virus infects the cell. And again, keep in mind, things here are not drawn to scale. So just look at how it is here. One virus goes in, takes over, things start to get made that are just virus. Well, it turns out every one of our cells has within it the ability to take any proteins that are being made, transport them into the ER where they get chopped up. Once they get chopped up, they get put into major histocompatibility complex 1. Complex 1 then traffics to the plasma membrane. Now, these proteins, these epitopes that are being put there, it's random. Could be self, could be non-self. Here, it's viral. Other times, if it's not viral, the cell's not infected, it's going to be self. If it's a self, T cells will ignore it. But here, they're not going to be ignored. CD8 cell, cytotoxic T cell coming along, interacts T cell receptor to MHC class 1. As we're going to see in a later chapter, T cell, TCR, MHC interact first, and then the next is, can the T cell receptor actually interact with whatever epitope? If it can, it's going to hold, and then the CD8 is going to come along and it's going to interact with the cell itself. Boom. Tells the cell time to die. Cell now kicks off apoptosis, cell suicide, and part of that is chopping up the nucleus. But what if it's just a T helper cell? Well, same thing again. These get, something gets inside, gets chopped up, gets transported to the ER, loaded up onto an MHC class 1, goes to the surface. T cell receptor interacts with MHC, If that can work, then the T cell receptor can interact with the epitope. If that's a yes, CD4 interacts, signal sent over, new cell abilities unlocked. Same with a B cell. B cell gets something presented to it by an APC, an antigen presenting cell, maybe a dendritic cell, maybe a macrophage and a lymph node. Doesn't matter. It gets it, loads it up on the MHC, along comes T helper cell. Again, TCR interacts with MHC, their interaction becomes stable, then can the T cell receptor interact with the epitope? If yes, CD4 interacts, signal sent over, B cell activates, either matures from immature to an effector cell, or if it's already been activated once before and it's just a memory cell, it starts going through mitosis and becomes a plasma cell once again. Now, when you step back and you look at it, there's going to be lots of different pathogens you're going to be exposed to on a daily basis. How our immune system responds to them is going to depend upon what they are and the state of your immune system. You get exposed to an infectious agent, a bacteria, virus, whatever. You have a normal response. Not only is your immune system going to kick in, but then it's going to have a memory. You're going to have protective immunity. Some innocuous substance, something that you should not be responding to, tree pollen, grass pollen, whatever, allergy. Under normal conditions, if you get some sort of an organ transplant, you should reject it within minutes. If you have a normal response to a self-organ, autoimmunity kicks in. If your cells haven't learned tolerance within the bone marrow. But if you have a deficient response, means something's lacking. Either CD8 cells cannot complete their job, or you're deficient in CD4 cells, which is one of the hallmarks for AIDS. You're not going to get protective immunity. You're going to get the infection over and over and over again. You'll never completely clear it. You'll never get lifelong immunity. You're not going to have to suffer from allergies if you're deficient in some way in your immune response. You're going to be able to get transported organs relatively easy. Turns out the anti-rejection meds that many patients get when they have an organ transplant actually instills an immune deficiency. So instead of having one genetically, instead of having one developmentally, they get one due to drug interaction. That's also how people have MS or some other autoimmune disorder. The drugs they give them that are supposed to help curtail their immune response basically gives them a deficient immune system that sets them up for recurrent infections.